젖소 농장에서 우결핵이 발생해 살처분이 진행됐습니다. 진안군에 따르면 지난 3월 진안읍의 한 젖소 농가에서 결핵균이 검출돼 젖소 100여 마리 가운데 4마리를 살처분한 데 이어 최근에도 17마리가 감염된 것으로 나타나 살처분이 진행됐습니다.
과천 서울대공원 동물원 일부에서 지난해 상반기부터 우결핵이라는 전염병이 유행한 걸로 SBS 취재 결과 드러났습니다. 동물끼리도 또 사람에게도 옮길 수 있는 이 전염병이 번지면서 서울대공원에선
We have to do two things at once. We have to bring this epidemic to an end, primarily by getting the vaccine out in large numbers to the entire world. And we have to make sure that we're ready because there will be another pandemic. And there's so many lessons about how we weren't.
Global warming is melting away glaciers at record levels, and due to that, the water level is increasing in the water bodies. Moreover, as the glaciers melt, it is also releasing the trapped viruses which were in the Arctic. And now the research finding coming up.
As shown in the video, it is essential to prepare for the early diagnosis of rare diseases, effective prevention of legal communicable diseases, and potential outbreaks of ancient infectious diseases in polar regions due to rapid global warming, which may cause a pandemic after COVID-19. To preemptively defend against these various diseases, XENOHELIX is developing molecular and nanotechnological diagnostic techniques. To achieve this goal, we are mainly focusing on two fields: circulating non-coding RNA analysis-based diagnostics and gene detection from ultra-small samples, including direct sequence analysis. Based on the technologies presented earlier, XENOHELIX has launched 16 items for research and diagnostic products which have been available from September last year. Within circulating non-coding RNAs, microRNA has been extensively researched for over 20 years because it is associated with many diseases and has drawn attention as a diagnostic biomarkers or cures. Despite over 120,000 articles have been published, not a single microRNA has been commercialized as a diagnostic biomarker. We believe that this problem is due to the efficiency of techniques for the purification of microRNAs and their accurate detection. First, we developed and launched products for efficient purification of microRNAs. Second, we developed GenoQ, a unique method for microRNA detection. It is a new, innovative technology with a completely different mechanism than the detection method from global companies. The advantage of our technology is that, not to mention of its accuracy, its simplicity, compared to other methods. Our technology is composed of only 3 steps, and it takes only 1 hour, while other technology consists of 6 to 7 steps, and it takes about 4 hours. Using the GenoQ method mentioned earlier, we aim to develop a Parkinson's disease diagnostic system over the past 4 years. We used a total of 640 clinical blood samples to identify Parkinson's disease-specific biomarkers in-house and performed a cumulative test of 1,100 using the GenoQ method. As a result, we developed 2 Parkinson's disease-specific biomarkers. We constructed a machine learning analysis algorithm and confirmed that the Parkinson's disease detection accuracy of the 2 biomarker was 84 to 86%. We are currently preparing to apply for KFDA approval as a screening diagnostic tool. Using blood microRNA biomarkers to diagnose Parkinson's disease can be the closest technique to an ideal Parkinson's disease diagnostic system, which is non-invasive, cost-effective, enables early diagnosis, and accompanying treatments. As mentioned in clinical laboratory news, we aim to automate the diagnostic system and believe we have reached 70% of that goal. As the second technology, we will talk about the detection of gene expression and a sequencing analysis of trace amounts of samples. We have developed a technology that can perform one-shot sequencing analysis of virus and their mutation in 50 samples within 6 hours and is in the process of patent registration. Real-time sequencing analysis is possible on site. We have developed a molecular diagnostic method for bovine tuberculosis in collaboration with Chungnam Livestock Sanitary Laboratory and the Jungang Baeksin. Bovine tuberculosis is a communicable disease, and all livestock farms are diagnosed by government officers who are veterinarians through simultaneous performance of interferon gamma test and intradermal test. These two techniques are used worldwide, and the market size is $1.3 billion. In Korea, about 1.1 million cows are tested annually, but the incidence of bovine tuberculosis is not decreasing, causing significant damage to livestock farmers. The problem is that the concordance rate of the two diagnostic methods is only 13%, and it is inconvenient for veterinarians to perform duplicate tests. The blood biomarker developed by XENOHELIX shows a 95% concordance rate with both existing methods through machine learning algorithm analysis, and its disease diagnosis accuracy rate is as high as 95%. Therefore, we expect to completely solve the problems of existing methods. We plan to conduct additional research to obtain a KFDA approval and enter the global market. As I mentioned earlier, the emergence of polar viruses due to rapid global warming and the infection of polar mammals and birds are already underway. We have developed a technology that can effectively detect these unknown variant viruses, and in collaboration with the Polar Research Institute, we have already developed a diagnostic system for poaching distemper virus. XENOHELIX is currently undergoing the application process for Parkinson's disease diagnostic products approval, with the goal of obtaining approval from the Korean Food and Drug Administration and being listed as new medical technology. Sequential development of liver fibrosis diagnostic system using the same technology is also in progress. At the same time, the company is conducting the final clinical study in preparation for applying for product approval for bovine tuberculosis diagnostic system. XENOHELIX is composed of 6 excellent researchers, 4 production members, and 5 management support team members, and is engaged in non-stop research and development as well as production and sales. In addition, the company is collaborating with several partner companies, research institute, and product suppliers, and is receiving close collaboration and advice on diagnostic system from Severance Hospital, St. Mary's Hospital, and Samsung Hospital. Starting in 2019, XENOHELIX has completed Series A funding of $5 million and is currently carrying out or finalized a total of $5.5 million in governmental projects, including a cross-ministerial medical device development of $4 million. The company has been recognized for the excellence of its technology, receiving the Minister Award twice. It has also completed GMP facility for medical device products manufacturing and obtained certification, and is currently producing and selling various products. XENOHELIX is dreaming of the future, preparing and executing, and is looking for partners to develop new technology for various diagnostics and therapeutics. Thank you for your listening.